Well, we've reached the final day of the Bioinformatics Week for the biotechnology program, and it's been a lot of fun to work with you on all these different topics that we've been covering. As I've mentioned before, if you take the next generation sequencing module, I'll see you again. If you take proteomics, I'll see you again. And if you take the special topic in clinical biomarkers, I'll see you then. But I have enjoyed this time with you, and I think that this set of topics has been a really interesting one to try to modify existing content and write whole new slides to cover. So thank you for this opportunity. Okay, so today our discussion covers biological pathways and networks. And I would state that a large part of this lecture is the theme that we should be cautious about producing laundry lists. If you have a whole genome assay and you come back with 50 genes that have been differentiated between your two cohorts that change when stimulated or versus when not, it's difficult for most biologists to follow up on information like that. If you give them a list of 50 genes that have changed, trying to interpret that biologically is tough. You know, are they suddenly going to have 50 grad students joining their group, each one assigned to one of those genes? Not a great option, generally. So being able to prioritize information that comes back from biological experiments, being able to see the patterns within these long lists of genes or proteins that come back from our experiments is actually quite difficult. And it's something that resources like biological pathways and networks can very much help us to do. So we're going to talk a little bit about pathways in general, what the promise of these would be, some discussion of one of the really big ones, Go terms. Many people will end up hearing about Go terms as part of their projects at some point. So those come from Gene Ontology. And then from there we'll talk a little bit about biological networks. Along the way we're going to have a few enrichment methods that we talk about. Specifically we'll be talking about WebGestalt as a framework for the use of over-representation analysis. And we'll also talk a little bit about Gene Set Enrichment Analysis. So I think it'll be an interesting talk for you to think about some things that don't generally get enough attention. I think everybody needs to have a little bit of literacy in biological pathways and networks. You don't have to specialize in it, but even a little bit of knowledge about what they offer can help you a lot. Okay, so I've tried to create kind of a broad map of what we're going to be talking about. As we work down the leftmost column on screen, we see classical biochemistry, microarray correlations, chromatin immunoprecipitation, protein immunoprecipitation, yeast-2 hybrid, confocal co-localization. These are just a few of the methods out there that enable us to say, how do two genes covary? How will the function of one relate to the function of the other? How will the location of one of these gene products relate to the produced gene product from the other? So classical biochemistry is kind of that stuff that you pick up in biochemistry class. Everyone remembers the Krebs cycle or citric acid cycle. We have pentose pathway, fatty acid synthesis pathways. We've got a huge number of these things, and they were worked out by very large labs that functioned over decades using techniques that would seem really quaint and old to us now. And yet, the biochemical lessons that they learned that are held remarkably consistent over a huge number of organisms is profoundly helpful to us. In several of the offices around here, I'm sure that you'll find a map of biochemical pathways that represents some microorganism or human cells for that matter. And these canonical pathways are very useful to our understanding any individual gene that we want to target now. So classical biochemistry has got a lot to offer us. Microarray correlation is another really important source of information. If you have the same cells analyzed under microarrays under 50 different kinds of stimulations, and you find that in exactly the same sets of stimulations this gene is up as another gene, and that whenever the other gene is down, this one is also always down, it becomes reasonable for you to say, I'll bet these two genes are being expressed at the same time suggests that they play part in a common process. So that's the microarray correlation approach. Chip-chip experiments, I think, are something you'll probably get exposure to, especially in next-gen sequencing. Chromatin immunoprecipitation to recognize interrelationships, well, among other things, of particular proteins that interact with particular genes that's worth knowing about. In that case, you have a cross-class association, right? We're not talking about proteins interacting with proteins there, but rather proteins interacting with DNA. Protein immunoprecipitation is one of the key ways that we figure out that this protein is associated with another one. For example, that they form a heterodimer. That would be the kind of information we'd like to get back from that. Yeast 2 hybrid is a massive system for understanding which genes are interacting with each other. Confocal co-localization. You'll sometimes hear people talk about fish in the context of confocal microscopy. This is a method where you tag protein 1 with a particular fluorescent tag and protein 2 with another fluorescent tag. You excite one of these fluorophores, it emits. That emission then excites the second fluorophore and it emits. So putting in the laser for the first fluorophore gets you a reporter on a different fluorophore. That's called FRET. Something like that is very useful if you're trying to demonstrate that these two proteins are found in the same neighborhood of the cell. So all of these are ways not about informing us about an individual gene or an individual protein or an individual messenger RNA, but rather about its association with others. And that's really what this lecture is all about. These larger collections, this larger view of understanding how genes operate in concert with each other. Okay, so that's experimental information that's coming in. Generally speaking, this is all going to lead to relationships. It's going to say to us that these two genes are part of the same pathway, for example. We'll talk about those relationships soon, because we're going to talk about the term ontology and what it means. So I want you to remember, relationships is half of that. The other piece of it is a controlled vocabulary. It's very difficult when different scientists are talking about the same biological process to reckon the results of one against those of another. One of the most common reasons, in the past at least, has been a predilection to use different names for the same gene. Right? So you might have 5 different synonyms that all relate to the same gene, but if you don't have your table of synonyms, you may not even realize that you should be looking at this other paper that relates to the same biological process. So having some ability to define our terms and to have everybody agree on what those terms should be is necessary for having a good ability to relate the results of one group to those of another. I mentioned just a moment ago HGNC, the Human Gene Name Consortium. This is a really, really big effort and a very important one, not only to say how many human genes are there and of what classes are they, but also to come up with a common gene naming system so we don't end up having this talking past each other problem that all the groups out there are using the same name when referring to an individual gene. That might not seem like it seems like something where you just sort of slap everybody involved and they settle on something, you know? But in fact, that process took decades to come to fruition. So a controlled vocabulary is a way to make clear what terms we're using to describe things. In this case, the gene catalogs themselves have required some amount of sorting of this type as well. Okay, so I mentioned that gene ontology comes from relationships and a controlled vocabulary. Canonical pathways are kind of the standard pathways that we think about. I've already mentioned a couple of these. So the example of glycolysis, right? What is this process by which we get pyruvate from sugar, right? Or from 6-carbon sugars. So that is an example of a canonical pathway. Now, people don't use the word canon all that frequently anymore. If you grew up in a Christian church, you probably have heard of some books called the canon and others called not the canon, or the Apocrypha, for example. This is a process by which, at the time, the Catholic Church, way back in history, had these big meetings where all the cardinals sat around and said, what are we going to treat as part of the Bible, and what are we going to treat as not part of the Bible? And so they had these big, long-standing arguments about things, and they finally settled upon these books that we say, okay, that is a Bible, anything else is not. And yet, if you look at different Bibles from different parts of the world, you will find that which books get included can be different. So it's we speak of this process as canonization, of setting a standard set of something. So a canonical pathway is to say, this is the canon, these are the biological processes everyone can agree on. And point-to-point networks represent a kind of different way of thinking about it altogether. So in a canonical pathway like glycolysis, I say, all right, I've got 6 carbon sugars in and I've got 3 carbon sugars out, so I need to I'm going to break apart glucose. I think everyone probably remembers that from a bit of biochemistry. So we have these steps that this follows, and that's all brilliant. But one of the things we note is that genes that are performing consecutive steps, one right after the other in these complex pathways, probably bear greater relationship than do ones that are quite far apart in these pathways. If you were to look up caffeine, for example, caffeine's mode of action, you would see a very complex and sprawling pathway, and things that are close together there, from a pathway perspective, are in the same pathway, just as things that are quite far apart, so long as they fall somewhere in the caffeine action pathway. And yet, from a network point of view, things that are much closer together in those diagrams probably bear greater relationship. So you can see that a point-to-point network assessment is actually a more valuable way, in some respects, to understanding biology. I would also note that many genes take part in many pathways, and as a result, understanding their actions in just one pathway, while ignoring all the other pathways they take part in, can be very problematic. So we're going to be comparing a lot of pathway versus network appraisals of systems. And they come up with rather different ways to look at biology, so I hope that will be useful. Now, from this, we're going to get several different possible outputs. Network modules represent sets of genes that are very closely related to each other. Random walk assessment is one way to ask how different things are. Cytoscape is a monster in this space. It's a hugely important piece of software that's free, that many, many people use for showing information from pathways and networks. Dimensionality reduction is a topic that we frequently encounter in statistics, but here it has quite a lot of meaning as well, that if you have a huge number of proteins you're trying to follow up, it's very helpful if you can cut that down to just a few pathways that contain most of those actors. So dimensionality reduction helps us to tighten up the set of questions that we're going to be asking of our data. Over-representation analysis and gene set enrichment analysis are two of the tests that we're going to talk about today. So this one slide tries to throw everything together in one hopper, and I hope it's useful for remembering just how all these pieces fit together. All right. Now, I spent an awful lot of time thinking about what people used to call functional genomics, or gene expression. That comes naturally because I'm a protein guy. So saying this protein changes substantially in expression level between these two cohorts is a really important kind of statistical question for us. But we see that people have been asking questions like this for a long time. Microarrays, for example, have been around since even before I was in grad school, and they've been providing a lot of gene expression information, not to just say which messenger RNAs are present, but at what levels are they expressed. So, although those methods have recently kind of been shoved aside in large part by RNA-seq, they're still quite valuable, and the methods used for RNA-seq analysis have a lot to do with what we originally created for microarrays. Proteomics is also inherited from that. So when you're evaluating how expression has changed between cohorts, you'll see kind of similar methods used. For example, this is called a volcano plot up at the top. In this case, we're seeing which proteins changed to a very low p-value up high in these plots, and things that are further left or further right are things that changed more in magnitude. So that's a volcano plot. Down at the bottom, we have a hierarchical clustering. Here we're not trying to say what's different here versus there, so much as we're trying to group together the samples that showed the most similar gene expression profiles. So we're reordering the samples across this top axis to put those that are most similar together. And we're trying to cluster together those genes that show the most similar patterns of expression on the y-axis. So we're clustering in both dimensions at the same time. That's a bicluster technically. Both of these kinds of analyses, though, tend to lend themselves to producing long lists of genes. So here we see that we have all of these different genes that are of potential interest because they show the pattern that our advisor told us to look for. That's not the scientific process, I'm sorry, I forgot. So yes, these are genes that correlate with our phenomenon of interest, right? So now we have a long list of genes, but we need to make sense of those. This is where biological pathways are so useful. So one way that you could think about this at the start is that each of the genes that you've put on your favorite protein or your favorite gene list have particular pathways that are associated with them. Some of those pathways are found among lots of genes, and others are found among very few. The greatest likelihood is that if many of your proteins that are interesting fall within a particular pathway, that's the pathway that interests you, even though there are multiple pathways for each of these. So think about turning this list inside out. Instead of saying, what are the pathways associated with these genes, we could say, what pathways explain the most of these genes? So here we can say, well, we had pathway 1 showing up for gene 1 and gene 2. We saw pathway 2 showing up for gene 1 and gene 3. We saw pathway 3 showing up for gene 2, gene 3, and gene 4, and so on. This gives you the ability to reorganize your data by which categories they fall within, rather than say what categories are represented among the genes we have. This is one of the ways that we do dimensionality reduction. Instead of 50 genes that we start with, we see that 3 pathways explain almost all of the data on our favorite protein list. All right, now why is it valuable to do this? We're going to start with better interpretability. Biologists love stories. I'm going to just say that if I randomly select 50 genes from the human genome and hand them to an experienced human geneticist, within 10 minutes, that human geneticist will have some story to explain how these 50 genes actually point to a very interesting biological phenomenon. Storytelling is one of our best human gifts, but it is a very dangerous thing to do. As I said, if I chose 50 random genes from the human genome, a human geneticist would be happy to tell me some story that relates them all. So that's something we have to guard against. But we still will be able to extract a lot more information if we have projected our gene list into a pathway than if we simply start from gene names alone. Having this relationship that potentially stretches over multiple genes that are on part of our lists is important for being able to understand what the biological phenomenon is, not just what is this one gene doing. Robustness really matters. So one of the things that we don't discuss enough is that when we do systems biology, whether we're doing RNA-seq or sequencing or whatever we're doing, we have lots and lots of hypotheses that we're going to be testing, and when we apply strategies like multiple testing correction to address that problem, we frequently find that things that really are differential are not producing great enough differences that our statistical test produces a really astronomically low p-value. This means we get false negatives. Genes that truly are different can't be called different because we have to be so careful from a statistical point of view. So false negatives are a problem. The other problem is that you may find some genes appear to be differential when they really aren't. You can get false positives. So you can imagine that this acts kind of like noise in your gene list. If you have 50 genes in your list and 10 of them are false leads, they're false positives, that's going to be noise that you need to resist as much as possible. By projecting these into the pathway space, you're better able to resist these erroneous claims of difference when they aren't, and also resist claims that things were not differential when they are. So this is a valuable thing. All right. And then we have sensitivity. So when people interpret things like volcano plots, they often use a lot of arbitrary rules. Things like a gene has to change its expression level by at least 2-fold up or at least 2-fold down, or we're going to ignore it, even if the p-value is significant. That's an arbitrary rule. It's not founded in the statistics at all. So when you see something like that, you may find that you throw out some very real changes. And it may be that if you're looking at each gene individually or each protein individually, you don't get a difference big enough to call it a real difference. But if you're looking at things on the pathway level and ask, are all of the genes in this pathway changing in the same direction, even if it's a relatively minor direction, relatively minor magnitude, you still have a better shot of picking it up if you're combining across more information to make that question. So to be very concrete about this, if you're computing p-values for individual genes or p-values for individual proteins, you may fail to pick up real differences that you would if you were instead computing your p-values for pathways or for networks instead of doing it per gene. So these are the kinds of things that we hope to accomplish by projecting into pathways when we're trying to figure out our favorite gene lists. Okay, so I see a wrinkled brow or two, so before we move ahead, are there questions people would like to ask? Good so far? Okay, good. So, where do we start? There's a world of different biological pathway databases out there. I tried to update this slide about 2 years ago, I believe, and I had to strip off about half the list and I had to add on a whole bunch of new ones. New systems for evaluating biological pathway information are showing up all the time. Some big things like Pathway Commons and Wiki Pathways, those things have been around for forever. We see systems like KEGG are so heavily used that they're just used all the time. Interesting names?
Yes.
Yes.
You know, they say we shouldn't trust Wikipedia, so
Ah! Okay, so yeah, they say you shouldn't trust Wikipedia because people like me have modified entries in there. Yeah, that's right. My wife was a high school teacher until last year, and she always had students fill out information from internet sources and intentionally made wrong information available in a particular Wikipedia entry to see how many of them would report it. And yes, they totally do. So yeah, is that mean? Yes, but did the students remember that? Yes, they did. Okay, so yes, things like Wiki Pathways are not part of Wikipedia per se. The people who alter Wiki Pathways are people who care about biological pathways. Generally, they're not entering a lot of false information. Now, is it possible that some editors of Wiki Pathways have considerably more liberal notions about what the truth is than others? Yeah, sure. We're human beings. And a graduate student who has spent his or her 5 years tackling away on a particular protein is going to have very strong opinions about what that particular pathway looks like that may disagree with what everybody else says. Right? So you can have people who are very self-interested. Well, I just published a paper saying this protein does this, therefore I couldn't possibly be wrong. Have to be careful about stuff like that. In any case, all of these different pathways have different emphases, but they all have useful stuff. So we have a couple different images that we've grabbed from BioCarta up here. That was this top one. This one comes from KEGG over here. We'll take a closer look at that later. But as we look across these different pathway resources, we should resist the notion that it doesn't really matter which one you pick and how can you know which one is right anyway. Because when you do that, you will see that different databases have different coverage. Some, for example, may include Arabidopsis thaliana, and others may not. If you're a plant biologist, and you're missing like your canonical species that we understand as well as any of them, that's a worrisome factor, right? So limited coverage in terms of which species are included and how up-to-date they are with various information. Some of these projects were created by graduate students or by postdocs, and we have to ask, is there currently someone acting as a curator for that system, or has it not been updated since 2013? These are very real things to worry about. And just the amount of link rot we get. You know, if you look at a paper from 2013, what do you think the odds are that the URLs in that paper are still live? It's not very good. I have a number of papers I published as late as 2015 that made reference to the Fenchurch MC Vanderbilt EDU web server. Well, when I moved away from Vanderbilt University, Fenchurch got packed into the packing crate and it's now here in South Africa. It's not at Fenchurch MC Vanderbilt EDU. So papers I published in 2015 didn't even have legit URLs at the end of 2015. Yeah. So we always have to do a little bit of detective work to figure out where these things have gone, how recently they were updated, how well they agree with other resources. It may be that the same information got coded different ways in these databases, or that this one accepts information from this source and the other one does not. A great deal of inconsistency. And the other problem that we frequently find is that different pathways are treated as different pathways, that this is an apple and this is an orange. And there's no relationship to show the outputs of this pathway flow into the inputs of this other pathway. So that kind of information can be rather limiting to understand these. Now, Gene Ontology, or GO, is so widely used that you have to get some sort of background in it. I just absolutely insist on this one. But first, before we move on to Gene Ontology, has anyone heard the word ontology before? Good. Some people have, some people haven't. All right, so ontology is actually a phenomenon that comes to us from philosophy. It's a theory about the nature of being, or the kinds of things that have existence. That's a pretty big statement, isn't it? If you're creating an ontology, you're trying to categorize all of the things within some scope. In this case, Gene Ontology is trying to capture all the different kinds of gene activity that we know about. That's a very, very tall claim, something that takes a lot of effort to accomplish. So in biology, our ontologies generally feature a controlled vocabulary and defined relationships. So what do those look like? In gene ontology, you have not just one relationship among all the genes, among many of the genes, I should say. None of these are comprehensive, covering all genes of any one organism. You have, in effect, three different organizing principles. So you have three different ontologies for the price of one. You have molecular function, biological process, and cellular component. So if you were looking at dopamine regulator, dopamine receptor D2, you see that its molecular function is a receptor of dopamine. That is its activity, what it's carrying out. Biologically, from a larger scale perspective, we say that it's part of synaptic transmission. Where is a synapse? What's that? In the brain, okay. More from a cell perspective, where would I find a synapse?
Between neural cells.
Between neural cells, okay. Yeah, so typically we have an axon that's sending a message and we have dendrites that are receiving it. The synapse represents that union where the two almost touch, basically. So it is this transmission between pairs of cells. So dopamine gets released by one cell and its reception causes a change in the cell that's receiving it. So we see that from a cellular component perspective, it's located in the plasma membrane because this is the synapse, it's the boundary of one cell and the next. But these three different assessment strategies mean that GO is kind of three hierarchies in one. It's going to show where something is located, the large-scale biological processes involved in the molecular function. Now, you'll often see several kinds of relationships tied to these. And I think these will seem common sense, but over time, these lists have changed very slightly and how they get defined has changed. So is one thing. So let me see. David Tabb is a primate. Can we accept that statement?
Yes.
Okay, thank you. I appreciate that. Thank you for including me. All right. Now, let us now flip that around. We're going to test the opposite. A primate is a David Tabb.
No.
No. All right, so we already know that commutative property is not going to work on this. Is a typically puts an individual into some group identity. Part of is one of these ways that we need to specify that there may be multiple types of components, and this is one of those components. Has a part is kind of the opposite of that relationship. Regulates is obviously one that shows up all over the place. So we have all kinds of these relationships taking place. So as we sketch this out more broadly, we see that we can make these different relationships at a whole different series of hierarchical levels. So at the top, we have biological process. You remember that was one of the three big hierarchies. Within that, we have single organism process and cellular process. As part of that, we have single organism cellular processes. Cell death is the more specific example of the single organism cellular process. And down at the bottom, we have programmed cell death. Do people know another term for programmed cell death? Apoptosis, right. Okay, so we can see then that there's a hierarchy here, and that all of these are represented by different terms within the gene ontology. And this is just one particular traversal down the tree. This is a very broad tree with hundreds, if not thousands, of such boxes. In this case, we have a plasma membrane, intrinsic component of the plasma membrane, an anchored component of the plasma membrane, an anchored component of the external side of the plasma membrane. This is the kind of hierarchy that we build at multiple stages through Go. All right. Getting information into Go is a very complex process, and they have full-time curators that take on this task. Not every database can say that. So we see that an awful lot of the annotation that makes it into Go has done so through automated means. Software says these two proteins or these two genes are related, and that's how it made it in there. We also have manual annotation. A particular curator has said, this new paper has come out that shows that this gene is associated with this function. But all of the annotations are labeled with a where did we get this information. So there may be a PubMed number to tell you this particular paper implicated this gene in this process. What kind of evidence was used to support this relationship is also there. So we see that there's a huge number of different codes that can be used for this. I'm not going to go through each of those, but you can see that they do separate out the classes of experiments that were involved, whether this was a computational thing or not. Was there an email from somebody to say you really need to put this protein in that process, etc. All right. So how much information is in Go? First off, how many genes do we have in humans? How many protein coding genes? What's that? A lot. Okay. When I was in grad school, there were some people who were still saying there must be 100,000 genes. Humans are so complex. Is that a good number? Not a good number. Okay. You'd go lower?
No, isn't it too low?
You would go more than 100,000 genes? You would be pretty lonely on that one. Most people would say humans have fewer protein coding genes than 100,000.
Oh.
Yeah. It was on screen just a moment ago.
No, it wasn't.
All right. Here, I'm going to pull this back up here. I'll just pull that off screen for just a moment. There we go. Oh, I snuck one past you. I had something on screen and I meant it to be there. Yes?
Is that like, they speak of junk DNA, so would this be like the not junk DNA?
Junk DNA is a pretty old term at this point. Most people would now argue that even if stuff doesn't code for protein, it has a lot of meaning in how the genome gets interpreted or regulated. So I would be careful about saying the term, because at this point, projects like ENCODE have found that a huge amount of the genome gets expressed regularly, whether or not it's destined to become a protein. Okay, but up here, we have a total number of genes with protein products This is the official number of HGNC right now, 19,194.
So is it ever the case that some of the junk DNA may code for protein in other species instead of that particular species?
Well, junk DNA is typically conserved somewhat less than the information in protein coding genes. So the non-protein coding DNA that my genome contains is different than, say, chimpanzees. There are some pretty huge changes in regulation that separate us from chimpanzee DNA. So if I were to walk you through all of this, it would probably lead us in a direction other than where the lecture is going. But I would note that out of our 3 billion, am I thinking correctly, 3 billion nucleotides, we have around 20,000 protein coding genes. We have a fair number of other things. You see this? Pseudogenes? 13,000 pseudogenes. We have almost as many pseudogenes as we have real genes. So things like ALU sequences are capable of producing products that are that look a lot like our own genes. Most of those have been turned off by regulation within our genomes. Okay, so I'm going to go ahead and return to my slides. Okay.
Question.
Yes, there's a question.
So these pseudogenes, so they produce products that are similar to our normal genes, but are they functional, or are they just chilling?
Oh, gosh, you really want somebody who's a proper geneticist to talk about that, I think. But
Okay.
I would say that many of these pseudogenes have the capacity to make more copies of themselves. So some of them do that through protein products, but I think generally you have stuff like retrotransposons. Have you heard that term? Okay, so these have the ability, if allowed to be transcribed, to integrate more copies of themselves into the genome. The way that I come to think of it from a kind of old book on the subject is that over the process of primate evolution, there was a bit of a war going on in our genomes with these pseudogenes making more and more copies within our genomes and threatening our the viability of the organisms, really. So for evolutionary purposes, the cells adopted things like DNA methylation as a way to prevent the creation of more copies of these pseudogenes. So it's kind of been fought to a standstill that we have all the evidence of pseudogenes throughout our genomes, and yet they're pretty inactive now because they've been blocked from making more copies of themselves. I probably made a geneticist roll over in his or her grave just then, but yeah, the process of evolution for humans has led to having an awful lot of DNA that is nominally not doing anything. But as I said, ENCODE established that there's an awful lot of it that gets transcribed anyway. And you have to think transcription is an expensive enough process that it wouldn't be taking place if it didn't have some biological role to play. Okay, why did I ask how many genes humans have? Because it's useful when you contextualize this. If you have 20,000 genes, you might think to yourself, why, there could only be 20,000 annotations for Homo sapiens. But in fact, we have more than 400,000 at the time I made this slide. So this is one of the things I want you to think about with pathways and networks. We're no longer talking about individual genes having an annotation or not. An annotation most typically is linking one gene to others. And there are far more handshakes possible in a room containing 20,000 genes than there are individuals. We talked about the cocktail party problem. How many different handshakes can you have if you have 10 people in a room, if you have 11 people in a room, and so on? The number rises more or less with the square of the number of people in the room. In the same way, the number of interconnections among genes rises at an incredible rate when you're able to talk about connections among different genes. Now, if you were doing chicken research, Would you be sad about this plot that I'm showing, or happy? Which one's the chicken? Ah, our friend Gallus. Okay, chicken. So is chicken well annotated? Chicken could be better annotated. What about this melanogaster thing? Anyone got a melanogaster? You know, if we set a bowl of fruit out, after a while we'll have some melanogaster. Okay, Drosophila melanogaster. How about Dana rario? Zebrafish. Ah. How about C. elegans?
What's that?
Anyone got worms? I don't want to know. I don't want to know. All right, it's a development, a model of development. There are a lot of different interfaces that you can use for this. I've already mentioned Bioconductor as a really, really valuable set of libraries available for R to do bioinformatics. Bioconductor has a toolkit that allows you to look up, for example, human gene names in Go and get back their biological process, their molecular functions, or the cellular component. Yes?
So what is the blue part and the orange part?
I've forgotten since I made this slide, but the point that it would make is relevant to the next slide. Okay, so where's all this information coming from? Now, across the bottom here, we have species. You can see that we've got Homo sapiens for these first couple. We have, the first three. Now, why would we have 3 mentions of Homo sapiens on this plot? Because Go is 3 different hierarchies, right? Molecular function, biological process, cellular component. Well, that's exactly the sort of thing I would ask a quiz question about. I don't even... The thing is, once I say, well, I would ask a quiz question about that, I feel like I shouldn't put it on the quiz anymore because I just told you all it's going to be on the quiz. I lie all the time. I'm a teacher. No, I'm kidding. Don't quote me. Okay, that's going to be just clipped right out of you.
Please us.
All right. So Go is three hierarchies in one. Each organism here shows up in 3 because all 3 hierarchies are represented for each of these species. So if you look at the human bars, these first 3, I see a lot of purple and a lot of green. So I could look up here to say purple is experimental evidence. Someone publishes a paper saying we use microarrays to prove that your favorite gene is involved in your favorite process. Boom. That's experimental evidence. It gets logged into the database. That record is updated to reflect that. What about green? Author statement. Some grad student phoned in to say, we just published a paper saying blah, 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 blah. Sometimes that happens. We've got brown. Similarity evidence. Similarity evidence. So maybe somebody did research in mice on an orthologous gene and found that it does X. So by similarity, because it's the ortholog in humans, we copy that annotation to humans. But we log every one of these changes to reflect why that change was made. Yes?
Is that why you, I don't know if my question is valid, but like, is that why you use mice to test a lot of the products on before you use them on humans because they have similar orthologs?
Well, it's also true that if you use grad students to test your drugs and stuff, people get all kind of upset about the ethics of it. If you have a drug and you test it on students, that's rude.
On students?
Yeah, testing on students is bad. Okay, so generally speaking, if you're trying to say, gosh, what would happen if we knocked out this gene in humans, you're not going to try passing that through ethics, right? Yeah. Instead, you're going to say, the ortholog for this is in mice, we're going to try knocking this out in mice, and then they can evaluate whether that is a legitimate thing. Because generally, making knockout babies, bad. It's bad. So yes, a lot of what we know about the human genome, about the human genes, human proteins, has been transferred over from other species, model organisms. Great. Okay, so we try to keep track of how stuff got into these databases because it can have pretty big impacts if it's in fact wrong. Okay, now I've talked enough about Go, Gene Ontology. Now we're going to talk about KEGG, the Kyoto Encyclopedia of Genes and Genomes, if I recall correctly. It may have changed what that means. All right, now I've circled a molecule here in red. Can anyone make out what that says from a long distance?
Caffeine.
Caffeine, very good, yes. Tea for everybody. All right, so here we have caffeine. Caffeine has biosynthetic pathways that lead to it, and it has biosynthetic pathways that lead away from it. So its next door neighbors are things like paraxanthine, theobromine, and 1,3,7-trimethyl uric acid. Perfect. Now, I have these little sets of numbers next to it. Like here I have 2.1.1.1.160. Is that an IP address? Does that give me a web page that tells me more about that process?
No.
No. So what are those dotted quads? What are those sets of 4 numbers?
Like classification or something.
Very nice, yeah. Enzyme Commission numbers, I think, is what they stand for. But yeah, it's an old school system that we use to name enzymes carrying out a particular process. So if you have a theobromine to caffeine reaction that gets catalyzed by a particular enzyme, it would be given the same Enzyme Commission number in humans and in chickens and in whatever species it could be found in. That's one of the useful ways that we can map across species what these different processes are. Okay, so KEGG has this way then of representing these genes as part of these interchanges of metabolites. Very useful. Okay, now WebGestalt is pretty cool. A lot of people feel that they can't do their biology unless they can buy a copy of Ingenuity Pathways. Anyone heard of IPA or Ingenuity Pathways software? It's pretty cool. It's very nice software. It's also kind of expensive though. And so it's very valuable to know about a few free web resources out there that can do a lot of what Ingenuity Pathways can do with a slightly less nice interface. Don't tell Bing I said that. Bing's a friend of mine. So WebGestalt is a tool that he created back when he was a postdoc. He's now been a professor for more than 10 years. But he's got this really great toolkit that enables us to accept ID types of a great many varieties and translate those into pathway information that the software has stored. Now, it may be weird to talk about identification types, but this is an important informatic problem. If you've used a microarray to measure a particular gene, you typically don't have the gene name as the label of that address on the microarray. Typically, you have a probe name instead, because you may have multiple probes that all correspond to the same gene, but they represent different sequences that are getting checked in the microarray. So you're probably going to have probe names as an input to this. And being able to translate those to gene names is a really important step. Frequently it's the case that if you import data from say a RefSeq database search of proteomics data, or a microarray from an Agilent version 6 microarray, that you're going to have a pretty big failure rate when you try to transfer that information in. And one of the very nice things that WebGestalt can do is tell you how many of the symbols that you imported did it actually recognize. Very, very useful. Okay, so importing the data into WebGestalt is very valuable, but it also supports a huge number of statistical analyses on top of that, and a fair number of different species. I believe that tuberculosis is still not on that list, but I need to check. Okay, so being able to look at gene ontologies, at pathways, at network modules, diseases and drugs, and chromosomal locations is all very useful kind of assessments. We're going to use this as a model for over-representation analysis. All right. Now, the user has provided some information here. They've given a list of proteins that they feel are different. Maybe this is from microarrays, right? So I have this list of genes now, 581 of them, that I claim are different. First off, is that a huge fraction of the human genome? 581. So to one approximation, we could say that's a half thousand, right? Mostly, right? So, and if there are 20,000 genes, we can say our difference list contains 1 in 40 genes of the genome. That's already a pretty big list. Pretty big list. All right. Now, development is a particular pathway that in this case contains 1,842 genes. So we're now going to compare our list for development versus the differential list that we provided. What comes out? So when we count the number of genes that show up on both lists, we see that 98 of them show up on both. So the question we ask is that higher or lower than we would expect by random chance? So in this case, we can try to make an estimate of how many of our genes we would expect to be on this list of 1842 by random chance. We already said that we had 581 genes. So if you were to choose batches of 581 genes at random from the whole genome, what number of them would you expect by random chance to intersect with development? So you can compute that number as well. So having observed 98 were on that list, and having expected 65 to be on that list, we see that 98 is bigger than 65. But is it a lot bigger, or just kind of inconsequentially bigger? So for that, we can use the hypergeometric test. So given a total of m genes, the number that you claim to be, oh, everyone freaked out as soon as I showed an equation? Is that what happened?
Yeah.
Oh!
We don't like that.
Oh, you're killing me. All right. What is this symbol?
Summation.
It's summation. Very good. That's a sigma. It means add up all of the. And then we've got j over I inside the parentheses. Do people remember what that represents?
No.
That is a combination. Essentially, how many sets of size I can you draw from j, if I remember correctly? All right. So this equation is going to help us to compute a probability that this would have happened by random chance alone. Okay, so given a total of m genes, in this case 581, where j genes are in the functional group, we think that number was 90 some, if we pick n genes randomly, what is the probability of having as many as we saw, k or more genes from that group? So this is the same thing as what we call a contingency table, which allows us to compute these probabilities directly. You can see how this works out in the 2005 paper. But just to say that the number is more than we would expect is not a statement about a p-value. We really do like to be statistically rigorous to say this is unlikely to have happened by random chance. And for that, being able to compute a little bit of math is actually a very good thing. You don't have to know the equation in order to run it on WebGestalt. It takes care of the computation for you. But it is important, I think, that you have some conception of where those values come from. As a bioinformaticist, I have argued pretty stridently against black boxes. You don't just hit the button and the software says X and you say, that's obviously a really great number, right? You need to have some notion where it comes from. Yes, sir?
Because I must know this for the test, this equation.
You didn't just ask that.
Just
If I say the hypergeometric test, am I going to expect you to work these numbers on the quiz? I am not. I'm not going to give you the numbers m, I, j, and n and say what's the probability that pops out. That's not going to happen. To understand that the hypergeometric test has something to do with overrepresentation analysis would probably be good. All right. Go is back. Look at that. There's Go. Way, way up there in the corner, we have biological process. See, I'm becoming South African. I said process. Here we have molecular function. Here we have cellular component. So here at the leaf of one of these trees, we have this, in this tiny little box, we have response to unfolded proteins. 12 genes were observed here, and the p-value that fell out of this was 1.32 E minus 8. So what does E minus 8 mean? If you were, we have a little decimal point here. Are we going to move it left or right to turn that into an ordinary number? Left. Right. So if this were E minus 1, we could move the period just one place to the left, the decimal point just one to the left, to give us 0.132. But it's minus 8. So that means that our value is decimal point 0.0000 0,0,0,132. That's our probability. Is that a very likely outcome?
No.
Not really. Right. So we can then compute these probabilities for each of these. But I want to show you a problem. If you treat all of these probabilities as being completely independent from each other, you've ignored something really key about Go. The same proteins that are causing this one to light up with a very low probability are also contributing to low p-values for the boxes above it. Because remember, those contain all of the genes below them as well. So we need to be careful that we don't say, well, this is significant, and that above it is significant, and that above it is significant too. This is a correlation among the probabilities that we can compute for individual processes. And there are clearly quite a lot of processes. This is not the full diagram. This is just those with significant p-values. There's hundreds or even thousands of these probabilities we're computing. So your chance of randomly having produced a low p-value is quite likely. Okay. In the end, we often try to portray the outcome of these by coloring in some of these diagrams. This is another one from KEGG. I think everyone probably recognizes the look. And we see that on the list of genes that we call differential, we had a fair number from this particular pathway. And this pathway was flagged as differential by overrepresentation analysis. And we see that these little blue boxes show, on a gene-by-gene basis, how this one got triggered as a meaningful hit. Okay. You can also use WebGestalt to do network-based analysis. I feel like I'm running out of time, though. What's our current time?
5 past.
5?
5 past.
5 past. Okay, so I've already run an hour. All right, I'm going to skip past here. All right, so why is overrepresentation analysis challenging? Well, thresholding can be quite arbitrary. It can be difficult to know of all the very low probabilities that we've got, which ones we want to hang our hats on. Ignoring the order of genes can be problematic. We don't, there's nothing about this pathway analysis to say, this gene is absolutely essential to this pathway, while these others are much less important. So that kind of, it's in the pathway or it's out analysis is kind of weak. Treating pathway genes as a set ignores that some of them are quite key to the functioning of that pathway. And others are quite promiscuous. You find many genes that show up in lots and lots of pathways. Gene Set Enrichment Analysis is another very, very big deal. It was produced by Joel Messeroff's team over at Broad Institute. I always struggle, I have to say, to try to explain its operation, but Gene Set Enrichment Analysis has become almost de rigueur. It's almost expected if you're going to do microarrays as an input, so it's a very important one to talk about. If you imagine all of the different genes that had any variance at all in your microarray experiment, you have two different classes of samples here, a cohort versus cohort comparison. And now you want to know, is this particular pathway showing up more frequently than you would expect by random chance alone? So one way to think about that is that you've sorted the proteins from those that are most up in cohort A to those that are most up in cohort B. In the middle, you've got this indifferent stuff, right? So we now have a ranking of proteins from, of genes, either way, to things that are most up in A to things that are most up in B, sorted from top to bottom. Imagine that a particular pathway has an awful lot of genes that are close to the top of your up in A set. Not all of them, but some of them are. So if you were walking from your top gene on this list, those that are most up in A, you would disproportionately hit genes as you moved step by step that were part of this pathway of interest. And so its score would rise, 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 rise. Then you would start hitting genes that had nothing to do with this pathway, and its score would fall, 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 fall. That's this curve that you're seeing right here. So this is an example of a pathway that is very enriched in genes that are near the top of this list. And this maximum deviation tells you, this is the set of genes that give us the least likely random hits on this pathway. So it's an approach that doesn't require you to say, these genes are differential, everything else was not. You just need to be able to sort them in a way that gives you some association with one cohort over another, and it will capture that information. So Gene Set Enrichment Analysis is very valuable for that purpose. Okay. In interest of time, I'm going to skip past network biology. If you find this subject to be an interesting one and one you want to talk about, we can set up another time for you to just chat with me about it. So network biology, the biological networks point-to-point assessments will not be on the quiz. I'm going to include the slides though. Feel free to look up references all you like, and we can talk about them if you want. Scale-free networks, it's such an important topic. I feel so sad to skip past that. Cytoscape is amazing. You should download it and play with it because it's awesome. I'm just going to tell this story because it's one of my favorite things to mention. I went to the Department of Molecular Biotechnology at the University of Washington Med School in 1996. It was their yes, I'm old, thank you. It was the very first year that molecular biotechnology was enrolling students from the outside world. Before this, they kind of borrowed all of their students from biomedical engineering. It was a cool department. It was like a bunch of all-stars. We had like Lee Hood, who is like this massive figure in systems biology. Biotechnology would not be the same without him, let's just say. He was heavily involved in inventing the automated sequencer. I think people have heard of that, right? Lee Hood, big name. He had a whole bunch of all-stars that were creating the field of proteomics, the one that I joined. Anyway, neat department. They enrolled 2 students in 1996, and I was one of them. And I was so proud. I'm going to go to the University of Washington. I'm going to go be a Husky. It's going to be awesome. Didn't realize I was going to get made fun of for graduating from Arkansas. Anyway, the other student who they enrolled that year was a fascinating guy named Trey Eidecker. And Trey had just finished his master's degree, I think in electrical engineering, at MIT. Now, I was used to being a big fish, right? I was like, I am smart, I am awesome. I arrived and met Trey Eidecker and I said, I'm in a different league just now. It was a surprising thing. Trey, already having gotten his master's degree, had some notion what he wanted out of grad school. And he focused very strongly on problems of how you represent relationships among genes visually. He went off to become a professor at UC San Diego, University of California, San Diego, created Cytoscape in his very first year as an assistant professor, and his career has just been in flames ever since. He's an amazing guy. I just want to say, when you get to graduate school, when you start a master's or PhD program, if that comes for you, you may discover that although you have always been the big fish, you may encounter a bigger fish. This does not mean you cannot also be a success. I'm proud of what I've done with my career, but my goodness, I spent enough time comparing myself to Trey and feeling awfully short. Okay, Cytoscape. I realize that is not part of, that's not on the quiz, but Cytoscape, amazing software, totally worth downloading and playing with. Network distances, yeah, that's all good stuff. All right, we've made it to the last slide. Building your assessment on pathways or networks rather than individual genes and proteins will have 2 key effects. Number 1, your biological interpretability should be far greater by projecting into pathways by saying, how do these genes map to what's known about biology? You're going to understand your gene list far better. Number 2, you will incorporate more data in each statistical test. If you're doing your testing on the basis of pathways rather than individual genes, very, very helpful for you. Gene Set Enrichment Analysis and Overrepresentation Analysis are 2 of the most common statistical tests we use when we're saying, did we have more of a particular pathway in motion than we would have expected by random chance? Point-to-point assessments and graph representations of biological pathways we're just not going to discuss today. It won't be included in the assessment next Wednesday, but I really appreciate this chance to talk with you, and with that, I will see you at 11:30, where we're going to talk a lot about everyone's tool in statistics, the t-test. because I just don't want you to go out there unarmed. I want you to be ready for people to challenge your results, because it might be me, you never know. All right, thanks everybody.
Thank you.